Two communities in India carried the same impossible question for generations. Were their ancestors really connected to the ancient Jewish world, or had centuries of isolation turned memory into legend? One group lived along the Konkan coast near Mumbai. Another survived far to the south in Kerala. Both kept Jewish traditions in a country thousands of kilometers from the Levant. Both told stories of ancestors who had arrived from the west, and for decades, neither archaeology nor old documents could settle the argument. Then scientists began comparing their genomes, and the answer was not simply yes or no. It was something far more revealing. The DNA showed that India's Jewish communities were neither untouched descendants of one ancient migration, nor purely local populations that had adopted a foreign religion. Their genomes recorded a long, complicated history of migration, marriage, isolation, survival, and reinvention. But the most surprising part came when researchers compared different Jewish communities inside India and discovered that the same religious identity could hide completely different genetic histories. India is rarely the first place people imagine when they think about the Jewish diaspora, yet Jewish communities have lived there for centuries. The best known are the Bene Israel of Western India and the Cochin Jews of Kerala, while later communities such as the Baghdadi Jews arrived through more recent trade networks. Each group developed separately. They spoke different local languages, lived among different Indian populations, and preserved different traditions about where they came from. That alone should make us cautious about speaking of one single Indian Jewish DNA. There was never one founding event that created every Jewish community in India. The mystery is actually a collection of separate histories that sometimes overlap and sometimes sharply diverge. Genetics became powerful in this story precisely because written records were incomplete. DNA could not tell researchers what people called themselves, what prayers they recited, or what they believed. But it could reveal patterns of biological ancestry left behind by generations of reproduction. The Bene Israel became the most famous case. Their traditional homeland lies around the Konkan coast of Maharashtra, with Mumbai eventually becoming a major center of the community. According to oral tradition, their ancestors survived a shipwreck on the western coast of India. Different versions place that event at very different times. Some traditions push the arrival deep into antiquity, while other historical estimates place it much later. That enormous range is a warning sign for historians. Oral traditions can preserve important memories, but they rarely function like precise calendars. For the Bene Israel, the difficulty was made even greater because their early written history is extremely limited. What survived was not a continuous archive of documents. It was a pattern of customs, family identity, communal memory, and religious behavior that persisted even when outside contact with other Jewish communities became limited. One of the most famous details concerns the community's refusal to work on the Sabbath. Bene Israel families became associated with oil pressing, and surrounding communities remembered that they did not work on Saturdays. That practice survived as an outward marker of identity even during long periods when other elements of Jewish religious knowledge were reduced or transformed. They also preserved forms of the Shema and certain dietary and ritual practices. By the 18th century, contact with other Jewish communities and outsiders helped reconnect the Bene Israel with broader Jewish religious traditions. But that reconnection created a new problem. Outsiders could now compare Bene Israel practices with those of other Jews, and differences that had developed during centuries of isolation became reasons for suspicion. The question slowly changed from, how did these people get here? To something more personal. Were their ancestors really Jewish? Or that question became especially painful after many Bene Israel moved to Israel in the 20th century. Religious authorities debated questions of personal status and marriage eligibility, and members of the community sometimes found themselves forced to defend an identity they had never considered uncertain. The issue was not merely academic. It affected families, marriages, dignity, and belonging. Traditional history said there was an ancestral connection to Jews from the West. Skeptics pointed to centuries of life in India and the strong physical and cultural resemblance between Bene Israel and neighboring Indian populations. Before genome-wide studies existed, both sides could select pieces of evidence that supported their preferred story. Genetics was supposed to settle the question. Instead, the first genetic studies made the mystery even more confusing. Researchers initially looked at different parts of the genome separately. Mitochondrial DNA, inherited through the maternal line, produced one picture. Many Bene Israel maternal lineages were clearly Indian. That suggested that local Indian women had played a major role in the founding or expansion of the community. Y chromosome studies, which follow paternal lines, produced hints of connections with Middle Eastern populations. Some paternal markers resembled lineages found among Jewish or other West Asian groups. But those studies often relied on small samples or limited genetic markers. Autosomal analyses, which examine ancestry across the rest of the genome, were also difficult to interpret because Indian populations themselves already contained complex layers of West Eurasian-related and South Asian ancestry. A Middle Eastern-looking signal could be confused with older ancestry already widespread in India. That is one of the most important technical problems in the entire story. Modern Indians do not descend from one simple ancient population. The subcontinent contains some of the deepest and most complex population structure anywhere in the world. Different groups carry varying proportions of ancestry related to ancient South Asian hunter-gatherers, ancient Iranian-related populations, steppe pastoralists, East Asian-related populations, and other regional sources. Long periods of endogamy then separated many communities from their neighbors. That means a scientist trying to identify a specifically Jewish or Middle Eastern contribution inside an Indian genome cannot simply search for West Eurasian DNA. Some West Eurasian-related ancestry entered South Asia thousands of years before any historically documented Jewish migration to India. The challenge is to distinguish an old shared ancestry layer from a much more recent admixture event. In 2016, researchers finally approached the Bene Israel problem with genome-wide tools powerful enough to separate those possibilities. They genotyped 18 Bene Israel individuals and compared them with hundreds of people from dozens of Jewish, Indian, and Pakistani populations, along with wider global reference groups. Instead of relying on one maternal lineage or one paternal marker, they examined patterns across the genome. They used principal component analysis, ancestry modeling, identity by descent sharing, linkage disequilibrium decay, haplotype sharing, and other statistical techniques. Each method asked the question in a slightly different way. If all of them pointed toward the same population history, the result would be much harder to dismiss as coincidence. The result was clear. Bene Israel genomes resembled Indian populations strongly, as expected from centuries of life in India, but they also contained a substantial ancestry component connected to Jewish populations that was not found in the same pattern among surrounding Indian groups. When researchers tested combinations of one Jewish and one Indian population as possible proxies for the ancestral sources, many of those Jewish-Indian pairs produced statistically significant evidence of admixture. Pairs made only from Indian populations or only from Jewish populations did not reproduce the same consistent signal. This mattered because it showed that the result was not simply the ancient ancestry mixture shared by many Indians. Something more recent had happened. The genetic clock placed that mixture roughly 19 to 33 generations before the individuals in the study, corresponding in the researchers' model to about 650 to just over 1,000 years ago. The average estimate was close to 800 years. That date does not prove the traditional shipwreck story, and it does not identify the exact city or country from which the Jewish migrants came. What it does show is that a real admixture event between a population related to Jews of the Middle East and local Indian ancestors occurred during the last millennium. In other words, the central idea behind the Bene Israel tradition, that their community formed through contact between Jewish migrants and people in India, was supported by genome-wide evidence. Even more revealing was the sex bias in the ancestry. Researchers found that the Jewish contribution was stronger in a pattern consistent with more Jewish male ancestors and more local Indian female ancestors. Mitochondrial DNA was mainly Indian, while comparison between the X chromosome and the autosomes pointed toward a larger male Jewish contribution. That produces a very human reconstruction of the founding event. Rather than a huge population arriving and replacing local people, the genetic evidence fits better with a relatively small group of Jewish or Middle Eastern men entering an Indian coastal population and forming families with local women. Their descendants then became increasingly endogamous, marrying mostly within the community. That endogamy left another signal. Bene Israel individuals share unusually long stretches of DNA with one another compared with many surrounding populations. Researchers detected evidence of founder events and population bottlenecks after the original admixture. This is exactly what scientists expect when a relatively small population forms and then remains socially separated for many generations. Genetic drift becomes stronger, some variants rise in frequency by chance, and the community develops a distinctive genetic profile. This does not mean the population became biologically isolated in an absolute sense, but it does explain how a mixed ancestry event that happened centuries ago could remain visible today. The study did not prove every detail of Bene Israel oral history. It could not identify 7 shipwreck survivors. It could not confirm an 8th century BCE arrival. It could not say whether the founding men came from Judea, Yemen, Iraq, Persia, or another Jewish community connected to Indian Ocean trade. Genetic reference populations are modern or relatively recent groups, not perfect replicas of people who lived 1,000 years ago. The researchers therefore used Jewish and Middle Eastern populations as proxies. That distinction matters. DNA can reveal that an ancestral source was related to known Jewish populations, but it cannot reconstruct a lost passport from the medieval Indian Ocean. Still, the finding was powerful because it resolved the part of the mystery genetics was actually capable of testing. Bene Israel were not simply another Indian population with no unusual connection to Jewish groups. Their genomes carried a distinct, datable history of Indian-Jewish admixture. The same result explained why earlier studies had seemed contradictory. Maternal DNA looked mainly Indian because many founding mothers were local. Paternal studies hinted at the Middle East because more of the outside contribution came through men. Autosomal studies became decisive only when enough markers and the right comparison populations were used to separate recent Jewish-related ancestry from the much older West Eurasian-related ancestry already present in India. Then, researchers turned to the Cochin Jews, and the story became even more complicated. Jewish life on the Malabar coast of Kerala is documented far back into the medieval period. Later traditions push the community's origins earlier, sometimes connecting them to ancient trade routes or refugees from events in the Roman world. Kerala sat on one of the great commercial highways of the Indian Ocean. Ships from Arabia, Persia, East Africa, and eventually Europe arrived for spices and other goods. A Jewish presence there is historically plausible without requiring one dramatic migration event. Over centuries, different Jewish arrivals could have joined an existing local community. Genome-wide research on Cochin Jews found exactly the kind of layered history such a trading region might produce. In one major study, researchers analyzed 21 Cochin Jews and compared them with hundreds of individuals from Jewish, Indian, Pakistani, and global populations. Like the Bene Israel, Cochin Jews resembled local Indian populations strongly, but they also carried a considerable Jewish-related ancestry component that distinguished them from most neighboring non-Jewish Indian groups. Statistical admixture methods detected relatively recent Jewish gene flow, estimated at roughly 13 to 22 generations before the sampled individuals, corresponding to around 470 to 730 years in that model. The timing is important because it does not necessarily represent the first Jewish presence in Kerala. Historical evidence for Jews on the Malabar coast predates that estimate. Instead, the genetic signal may reflect later arrivals mixing into an already established community. Cochin Jewish history includes contacts with Yemenite, Sephardi, Middle Eastern, and later European Jewish populations. After expulsions from Iberia and changes in Indian Ocean trade, additional Jewish migrants reached South Asia. A genome can record one strong admixture pulse even when cultural history began earlier. This is another reason genetic dates must be interpreted as population events, not as simple founding certificates. One ancestry model in the Cochin study estimated a larger Indian component and a smaller Jewish component, around 79% and 21% respectively, but those numbers should not be treated as exact universal percentages for every Cochin Jew. They depend on reference populations, methods, and the sampled individuals. What matters more is the repeated result across several techniques. Cochin Jews carried both Indian and Jewish ancestry, and the Jewish-related signal was recent enough to distinguish it from ancient South Asian population history. Their mitochondrial DNA was again heavily Indian, suggesting substantial contribution from local women, although genome-wide tests of sex bias were less conclusive than in the Bene Israel. The Cochin genomes also showed high endogamy and evidence of recent bottlenecks. Once again, religion and social organization shaped the genome. This is a theme that appears repeatedly in both Jewish diaspora populations and many Indian communities. Marriage within a defined group preserves ancestry patterns that would otherwise blur rapidly. In India, where caste, tribe, language, geography, and religion historically created powerful marriage boundaries, a small Jewish community could remain genetically recognizable for centuries, even while living among much larger neighboring populations. But there is a danger in turning these findings into a story of genetic purity. Neither Bene Israel nor Cochin Jews are purely Jewish or purely Indian, and the science does not require either category to be pure in the first place. Jewish populations across the world have histories of migration and mixture. Indian populations do too. Identity survives through culture, family, law, memory, and community, not through an untouched genome. The real discovery was not that Jewish DNA somehow remained frozen in India. It was that historical encounters between populations left patterns strong enough to remain detectable after centuries of local life. This becomes even clearer when we look at another group associated with Jewish identity in India, the Bnei Menashe of Manipur and Mizoram in the northeast. Their story is fundamentally different from the Bene Israel and Cochin cases. Bnei Menashe members identify themselves with the biblical tribe of Manasseh, one of the tribes associated with the ancient northern kingdom of Israel and the long tradition of the lost tribes. Their modern movement toward Judaism developed in the 20th century among Mizo and Cochin communities, whose languages and regional ancestry are connected to northeast India and neighboring parts of Southeast Asia. The Lost Tribes story begins with the Assyrian conquest of the northern kingdom of Israel in the 8th century BCE. Assyrian policy involved deporting and resettling populations, and the later fate of many deported Israelites became the subject of enormous speculation. Across centuries, communities from Africa to Asia were linked, by themselves or by outsiders, to those missing tribes. But a claim of descent from an ancient population separated by nearly 3,000 years creates an extremely difficult genetic problem. Even if a small group of Israelites had somehow reached a distant region and mixed into a much larger local population, their genetic contribution could become too diluted or too genetically ordinary to identify confidently today. That is exactly why Bene Menashe should not be presented as a case already solved by DNA. Available genetic testing has not produced the same clear genome-wide Middle Eastern admixture signal seen in Bene Israel. Reports over the years have discussed particular maternal lineages or small studies, but no result has established a direct genetic descent from the biblical tribe of Manasseh. The absence of such proof does not disprove religious identity or personal belief. Genetics is not a test of faith. It only means that the specific historical claim of biological descent from an ancient Israelite tribe has not been demonstrated by the kind of population genetic evidence that supports Jewish-related ancestry in Bene Israel and Cochin Jews. That distinction has become increasingly important because Bene Menashe migration to Israel is not merely historical. In November 2025, the Israeli government approved a plan aimed at completing the immigration of eligible members of the community from northeast India. Official Israeli parliamentary information later stated that up to 1,200 people were expected during 2026 as part of a multi-year process, subject to religious and administrative procedures. In April 2026, an official Israeli government education publication reported the arrival of 240 Bnei Menashe immigrants and described a broader plan involving thousands more by 2030. Those are political, religious, and humanitarian decisions. They are not equivalent to a new genetic discovery. That difference is essential. A government can recognize a community through legal or religious processes, even when population genetics remains inconclusive. A rabbinical authority can make a determination based on tradition, conversion, or communal history without waiting for a chromosome to deliver a verdict. The Bene Israel story sometimes encourages people to imagine that DNA is a courtroom judge that decides who is Jewish. It is not. Jewish identity includes religious law, culture, ancestry, conversion, and communal belonging. Genetics can reconstruct biological history, but it cannot determine spiritual legitimacy. The comparison between these Indian communities therefore produces a more interesting conclusion than the headline, "DNA proved they were Jewish." For Bene Israel, genome-wide evidence strongly supports substantial ancestry from both Indian and Jewish-related sources, with a mixture event during the last millennium and a clear sex-biased pattern involving more Jewish male and local Indian female ancestors. For Cochin Jews, genome-wide studies also support mixed Indian and Jewish ancestry, including relatively recent Jewish gene flow layered onto a longer regional history. For Bnei Menashe, a direct ancient Israelite genetic connection remains unproven. 3 communities connected to Judaism, 3 distinct historical pathways, and 3 different relationships between identity and ancestry. The science becomes even more revealing when we understand how researchers can date these events. Every generation, chromosomes inherited from a person's parents are broken and recombined before being passed to children. When 2 previously separated populations mix, the first generation carries long blocks of ancestry from each source. Over time, recombination cuts those blocks into shorter pieces. By measuring how quickly ancestry correlations decay across genetic distance, methods such as ALDER can estimate how many generations have passed since admixture. Haplotype-based methods examine shared stretches of DNA and compare them across populations. The dates are statistical estimates, not exact anniversaries, but they allow scientists to distinguish an admixture event 800 years ago from one that happened several thousand years earlier. This was crucial in India because ancient West Eurasian-related ancestry is already widespread across the subcontinent. Without dating, a Middle Eastern-looking component in Bene Israel might simply reflect ancestry shared with many other Indians. The genome-wide studies showed that the Bene Israel signal had a much more recent structure. Researchers could compare Bene Israel with numerous Indian populations, containing different proportions of older West Eurasian-related ancestry. Those populations did not show the same consistent Jewish-Indian admixture pattern. That is why the result was persuasive. The argument depended not on one exotic genetic marker, but on the timing, distribution, and genome-wide structure of ancestry. At the same time, sample size reminds us to stay cautious. The influential Bene Israel genome-wide study included 18 community members. The major Cochin study included 21. Those numbers were sufficient to detect strong population-level signals because the communities were relatively endogamous, but they cannot capture every family history or every subcommunity. Later, in future studies with whole genomes, ancient DNA, and broader sampling could refine the picture. Science rarely closes a historical mystery permanently. It replaces vague possibilities with models that can be tested and improved. Ancient DNA could transform this field even further, but India presents serious preservation challenges. Heat, humidity, soil chemistry, and microbial activity often destroy DNA in human remains. Successful ancient genomes from South Asia are still far fewer than those from colder regions, such as Europe or northern Eurasia. For Indian Jewish history, this means researchers rely heavily on living populations. If archaeologists could recover genomes from securely dated Jewish burials on the Konkan or Malabar coasts, they might directly test how ancestry changed through time. Until then, modern genomes remain indirect records, reconstructed backward from descendants. There are also ethical questions. Genetic studies of identity can easily be misunderstood or weaponized. A community may feel vindicated when a study detects ancestry connected to its oral tradition, while another community may feel threatened when the expected signal is absent. But absence of a signal is not proof that a culture is fake. A religion can spread through conversion. A language can spread without mass migration. A small founding population can leave little genetic trace after centuries of mixing. A community can preserve a real historical memory, while the biological contribution from the original migrants becomes tiny. Human identity is always larger than a percentage on an ancestry chart. This is especially important in Jewish history because the diaspora itself was created through movement. Jewish communities formed in the Middle East, Europe, North Africa, Central Asia, Ethiopia, Yemen, India, and elsewhere. Some retained stronger genetic continuity with Levantine or Middle Eastern populations. Others absorbed much more ancestry from surrounding populations. Different communities experienced founder effects, conversion, persecution, trade, and intermarriage at different times. There is no single DNA sequence that makes a person Jewish. Population genetics studies shared ancestry patterns, not religious authenticity. The Indian case may be one of the clearest examples of that principle. The Bene Israel spent centuries being deeply Indian and deeply Jewish at the same time. Their language, food, appearance, and maternal ancestry became strongly rooted in Maharashtra. Yet their genomes still preserve evidence of Jewish-related ancestors from the West. The Cochin Jews became inseparable from Kerala's history while maintaining religious connections across the Indian Ocean. Their DNA similarly records both local and outside ancestry. These are not contradictory identities. They are exactly what we should expect when a diaspora community survives for centuries in a new homeland. One more detail makes the result especially powerful. The same genome that preserves ancestry also records isolation. When a small community marries mostly within itself, rare variants can become common simply because there are fewer founders contributing genes to later generations. Researchers detected this kind of founder effect in both Bene Israel and Cochin Jews. That matters medically because some inherited disorders can become more frequent in endogamous populations, but historically it also acts like a signature of community continuity. The population did not merely mix once and disappear into the surrounding majority. It remained socially recognizable long enough for drift, bottlenecks, and shared DNA segments to accumulate. In that sense, the genome records two different phases of the story: first contact, then preservation. The first phase explains why Jewish-related and Indian ancestry appear together. The second explains why that mixture is still detectable centuries later. It is a reminder that history is not only about people moving from one place to another. It is also about what happens after they arrive, whom they marry, which traditions they keep, which boundaries they maintain, and how those choices echo biologically across generations. A migration may last a few months. Its genetic consequences can remain visible for 1,000 years. That is why these communities became clear genetic archives of an older encounter across the Indian Ocean. And that brings us back to the original mystery. Scientists did not uncover one hidden Indian Jewish gene. They found something more historically meaningful. They found a sequence of encounters. A relatively small migrant population reached western India and mixed with local people. Local women contributed strongly to the formation of Bene Israel. The descendants became endogamous, preserving both sides of that ancestry. In Kerala, Jewish-related gene flow entered a community whose history was already tied to Indian Ocean trade and local Indian society. Those signals remained detectable because social boundaries preserved them. The most dramatic result, then, is not that science proved an ancient legend word for word. It did something better. It separated the parts of the legend that genetics can support from the parts that remain beyond evidence. Bene Israel oral history remembered an origin involving outsiders arriving on India's western coast. Genome-wide DNA supports a genuine Jewish-related migration and admixture event, though not the exact shipwreck. Number of survivors, or ancient dates sometimes attached to the story. Cochin traditions remembered long contact with the Jewish world. Genetics confirms real Jewish-related ancestry, while showing substantial local Indian contribution, and later gene flow. B'nai Menashe tradition reaches toward the lost tribes, but that specific biological connection remains unresolved. For centuries, these histories survived without laboratories. They lived in Sabbath observance, marriage patterns, family names, ritual memories, synagogue communities, food, songs, and stories repeated from one generation to the next. Genetics arrived late. When it finally did, it did not replace those memories. It gave historians another layer of evidence, and in the Bene Israel case, provided powerful support for an ancestral connection that had been doubted by outsiders. There is something deeply human in the fact that the strongest evidence was hidden not in a royal archive, but inside ordinary descendants. Every recombined chromosome carried fragments of events no surviving witness could describe. A marriage on the Konkan coast centuries ago. A Jewish trader or refugee who never imagined that his descendants would one day live in Mumbai or Israel. An Indian woman whose name was never recorded, but whose mitochondrial lineage survived through generations. Communities that became small enough for chance and endogamy to leave strong signatures in their genomes. The laboratory did not discover abstract percentages. It recovered traces of forgotten families. And the mystery is still not completely finished. Researchers do not know the exact geographic origin of the Jewish founders of Bene Israel. They cannot say whether one migration or several produced the community. Cochin Jewish genetic history remains layered, and different historical subgroups may preserve different proportions and sources of ancestry. B'nai Menashe ancestry remains a separate unresolved question. New whole genome datasets, better reference populations, archaeological discoveries, and perhaps ancient DNA could alter parts of the story again. But one conclusion is already difficult to escape. India's Jewish communities cannot be explained by one simple story of either foreign settlers or local converts. At least, for Bene Israel and Cochin Jews, the genome records both. Their ancestors came from different worlds and became one population through marriage, migration, faith, and centuries of survival. The Indian ancestry is not evidence against their Jewish history. The Jewish-related ancestry is not evidence that their Indian identity is somehow less authentic. Both are written into the same chromosomes. That may be why this mystery mattered for so long. The argument was framed as a choice: Indian or Jewish, local or foreign, legend or fact. DNA revealed that the choice itself was wrong. The people at the center of the debate were carrying both histories at once. After generations of questions, the genome did not uncover a pure bloodline. It uncovered a meeting, and that meeting changed the story forever.